Hello, my name's Ben, and I'm going to talk you through some work we've done on comprehensive quantitative phenotyping of physiological dynamics, and this is some of the stuff that our group enjoys thinking about and doing. And so the first problem that we're trying to treat is working with these types of physiological signals. So whether you stick a sensor on a heart or someone's brain or measure them breathing, you end up with these kind of beautiful and intricate measures of different physiological systems. And the question is, can we turn these measurements that we're getting more and more of these days into something that we can use? Like, can we improve a diagnosis? Can we improve treatment? Can we model and understand what's happening? So that requires us quantifying and using complex methods to try and understand the patterns that are hidden in these signals that might allow us to do the things we want to do with them. And lucky for us, there's a huge toolbox of scientific tools of very clever people who've thought about these types of problems from lots of different angles. So we have methods developed in economics, biology, physics, medicine, as I've shown at the bottom there. And all these people have thought about this type of problem, where you have a complex, time-varying signal that encodes some complex dynamics. In the economics case, you might want to predict inflation. In the biology case, as I'm talking about today, you might want to diagnose or find some biomarker. In physics, we might want to understand different types of stars from measurements of how they pulse and change. So all these different people have thought about this problem in different ways. And as a result, we have this massive toolkit of quantitative methods to extract structure. And there are different questions and examples of that toolkit on this slide. We might ask, how variable are the dynamics? Is there fractal structure in the dynamics? Are they predictable? Are they stable? All these questions have quantitative ways to answer them. So we have this massive toolkit of methods. And so when a problem comes up, here's a big data set. We want to answer one of these questions. Can I diagnose this difference? Can I help work out what the right treatment is for some condition? Then us as kangaroos need to solve this problem of how do you pick the right method. And so there's, very powerful modern choices that we can make using deep neural networks and flexible learning architectures like that, which are very flexible, but they require lots of data typically, and it's hard to understand them. And so what we want to do is take the second route, which is to draw on an interdisciplinary literature of these powerful methods that I just outlined. But there are so many of them, how do we pick from all these different methods from economics, physics, biology, etc.? And so the approach that we advocate for and have developed tools to do is called the highly comparative approach. And this is where we try and compare as many methods as we can and see which ones work for a given problem. And so the first step to do this is to have a very large set of methods that we can actually quantitatively compare systematically. And so we've shown the tools that we've developed for a single time series. These are HCTSA and the Python version called PyHCTSA. And here you can compare thousands of these different methods and find the ones that work well on a given problem. And then we recently also developed one for pairs, where there's similarly hundreds of different methods for quantifying how two different parts of a system couple and interact. And similarly, we can measure hundreds of these statistics and try and find the ones that are relevant for a given problem. So this gives us a kind of statistical basis on which to search all of the beautiful methods that exist out there in science and try to identify the ones that are relevant for our problem, that both give good performance and also point us back to interpretable theoretical literature that could help us to understand what's actually happening underneath the time series. And so this is what an analysis might look like. We have some collection of time series, blue and red, in the simple case of a binary difference, and this could be healthy recordings in blue and pathological recordings in red. And then we measure all of these thousands of different methods. Each of these shown as a single column in this matrix, and all of these different time series are rows of this matrix, and we get this kind of quite visually beautiful structure. And this is the basis from which we can do statistical learning. Are there any columns here that give high numbers for the blue and low numbers for the red? Well, that would be a very useful candidate metric to, for example, diagnose the difference between healthy and unhealthy blue and red in this case. And so we've done this a lot. We've used this to find new features to distinguish meditators from non-meditators. We've used it to diagnose congestive heart failure from heartbeat series, to classify seizure, to try and track the distance to a critical point in noisy systems. This is an idea from physics, and we've able to calibrate that to a statistical measure for the first time and show that it tracks the mouse visual hierarchy, and lots of other examples down there in the bottom right of us or others using this approach. And to date, there have been hundreds of these types of examples. And same for the pairwise case, where we're trying to understand, well, what measures of coupling distinguish different conditions. So this is more relevant for multivariate time series, where we measure lots of different parts of a system at once. For example, in brain imaging, we might say, well, how strongly is this part of the brain coupling or communicating with another part of the brain? And we can find out, well, what's the best measure of coupling? So we do the same idea where we compare hundreds of different approaches to measure that coupling, and we find the one that works the best. So here are some examples of that in accelerometer data and different types of brain imaging data. And here's a representation of some of these hundreds of publications. I've just picked out a few from medicine, biology, and neuroscience of examples of people applying our method to their problems. And I try and keep a log of all of these. And then what the group's doing next is kind of digging into finding more nuanced ways to capture structure in these high-dimensional noisy biological signals. For example, tracking non-stationary changes across measurements of the brain that might help us better quantify sleep dynamics, finding underlying mechanisms in high-dimensional data. This is another useful problem where you don't often have the same resolution and sampling in all of the different types of dynamics you want to compare, and also comparing the quantity of time reversibility and trying to quantify that in non-equilibrium systems. So the take-home message is that, it's really important, if we want to tackle some of these big open questions in health, that we bridge the theory and methods from physics, statistics, mathematics, computer science, these types of quantitative disciplines, and properly embrace the health-related problems that need them through a two-way exchange. So this is my group, and thanks for listening.